Hey, all you cool adults and adolescents. Aren't you glad I didn't say cool cats and kittens? Um, Tiger King humor. Anyway, today's going to be the second lesson in vision. So we're going to call this video lesson 2B. And hopefully we'll have enough time to get through the rest of vision. So let's go ahead and get started. All right. So the optic disc is the part of the retina that is known as your blind spot. This is where the optic nerve and the retinal vessels enter and exit the eyeball. And since that is occupied by the blood vessels and the nerves coming in, there are no rods and cones there, so it makes your blind spot. And this is where we do a lab, and I may have to do a little different video on it. But in our visual lab, our vision lab that we do, we find our blind spot. And we'll talk a little bit about it. Its existence creates a necessary defect on the retina, an area where there are no cones or rods. And because of bilateral vision and quick involuntary muscle movements, it allows our brain to correct for this blind spot. And most people aren't even aware of it. The reason is the blind spot of your right eye does not overlap the blind spot of your left eye. So where your right eye is blind, your left eye can see. And where your left eye is blind, your right eye can see. And since with both eyes open, there's always at least one eye is getting all of the wavelengths of light for our visual field. Our blind spot goes undetectable for each individual eyeball. So to find it, you have to close one of your eyes. And I may do a quick little flip grid on how to do that on one of our Friday flip grids. The retina consists of two types of photoreceptor cells. They're called rods and cones. Rods are abundant in the periphery, so out away from the middle of the retina, where cones are found more frequently in the central areas. Each eye contains about 120 million rod receptors. They are adapted for low light threshold, high sensitivity, so they're good in low light. They don't work so well in bright light. They produce a low resolution, which means not very clear, and they are good for identifying black and white images. A loss of rods with age makes it difficult to drive at night. Okay, so one of the things that we do in class is we talk about how if you've ever been looking for something in the dark, you have a better chance of seeing it in your peripheral field of view than you do in your center, and that's because you're using rods out in the periphery. And I know I've been looking for things in the dark, and when I look directly at it, I lose it. But when I look next to the thing I think I'm looking for, I have a better chance of seeing it. Some of you may know what I mean. Cones, on the other hand, are just the opposite. They have obviously called cones because they have this cone shape on the back edge. They function in bright light to produce high-resolution color images. They exist in three varieties in the human eyeball, corresponding to the type of pigment they contain. So our cones are receptive to primarily red, green, and blue. Okay, deer do not have this cone. So not only do they have a low concentration of cones, they can't see red and anything in that spectrum. So that's why that hunter orange, that bright orange color, to a deer is not the bright orange color that we see. So that's why during gun season, when you go out and you wear your bright orange, the deer still have a hard time finding you, even though when you look at that bright orange, it stands out and is very easy to see. The photopigments are concentrated in the outer segment of the receptor, while the inner segment contains the nucleus and the organelles. And so you can see the nucleus there, and you can see the organelles and the mitochondria. And of course, we all know what the mitochondria does for the cell. Eye cavities and chambers. The lens is an avascular refractory structure situated posterior to the pupil and iris, which means it is behind the pupil and iris. It is obviously transparent because light passes through it. Again, we see wavelengths of light. So for light to get in your eye and get to the retina where the image is made, it has to pass through the cornea, through this fluid in the anterior chamber called the aqueous humor, and then it passes through the lens. When it passes through the lens is going to bend it slightly, and this is what's going to help us. Of course, it's also going to bend slightly when it comes in the cornea, but when it bends through the lens, this is what allows us to focus the light. It attaches to the ciliary muscle of the ciliary body by suspensory ligaments, those guys right there, that fine-tune the focusing of light on the retina. So when you go to focus on something, these ligaments will pull on the lens and cause the lens to either get thicker or get thinner, depending on if you're looking at something close or far away. The lens divides the eyeball into two cavities. The anterior cavity is anterior or in front of the lens, and the posterior cavity, also known as the vitreous chamber, is behind the lens. So the lens is a physical barrier between the fluid in the front and the fluid in the back of the eyeball. Okay, so again, this fluid that's in the front is called is the aqueous humor, and it fills the anterior cavity or anterior chamber. And the fluid behind the eyeball is called the vitreous humor, and it fills the vitreous chamber. And they don't mix because the lens separates the front from the back part of your eye. The eye cavities and chambers. The much larger posterior cavity of the eyeball, the vitreous chamber, lies between the lens and the retina, and its job is to keep the retina nice and pressed up against the back of the eyeball and keep the eyeball shape. Within the vitreous chamber is the vitreous body, also known as the vitreous humor, a transparent jelly-like substance that holds the retina flush against the choroid, giving the retina an even surface for the reception of clear images. It's kind of what I just explained. Occasionally, collections of debris called vitreal floaters cast shadows on the retina and create a spot in our field of vision. We call them floaters. They are usually harmless and do not require treatment. Given time, that will settle in the bottom of your eyeball and it won't cast that shadow on the retina anymore, and you don't have your floater. The aqueous humor, again, great name for a rock band. The eye requires a constant bath of nourishing fluid to deliver enough oxygen to support the avascular lens. Okay, so it's avascular, it has no blood supply, but it still needs oxygen. So if it doesn't have blood, and we all know from the cardiovascular system that our tissues get oxygen from the blood, how does the tissue get the oxygen? Well, I'm glad you asked. It gets it from the aqueous humor. It is constantly being secreted into the eye and flushed out and is replaced at all the time. It basically will be replaced every 90 minutes, and that is how oxygen is supplied to that transparent part of your eyeball. And it's a good system because blood vessels are not transparent. You really can't see through them. So having blood vessels in the way of light coming in your eye would hinder your visual ability. So this is a good way to solve that problem. This need is accomplished through the production of the aqueous humor, which flows through the anterior cavity of the eye and is replaced every 90 minutes. Let's talk about the pupillary response, and this is when your pupil gets larger or gets smaller depending on the amount of light. The pupil is an opening in the center of the iris. It is composed of a radial muscle that radiates away from the center and the circular muscle that is in the center. So these are your radial muscles, and these are your circular muscles, and these are your radial muscles that run this way. Contraction of the inner circular muscle causes the pupil to constrict. So this is what happens when you have a lot of light. The pupil constricts to limit the amount of light that gets in. And then when you're in low light, then the radial muscles constrict, which opens the pupil and lets more light in. So the larger the hole, the more light will get into your eye. And of course, we see that in low light situations. So what I want you to do is pause the video, go into your bathroom and close, hopefully a bathroom that doesn't have windows, and close the door and turn the lights off. And look into the mirror, try to find yourself in the dark, look right at yourself in the mirror, and if you can reach the switch, turn the light on. And when you turn the light on, you will be able to watch your own pupils constrict. You can't see them dilate because when you turn the light off, you're in the dark. You won't be able to see them. But if you do that in reverse, if you look at your eyeballs in the mirror when you turn the light on, you'll be able to watch your pupils constrict. So go do that now, and we'll see you when you get back. Well, we couldn't make it through lesson 2 in just 2 videos. So I'm dividing 2B into 2 shorter 10-minute videos. And so we'll have 2B and 2C, but both 2B and 2C will be one exit slip. So now that this video is over, since YouTube won't let me do any videos longer than 15 minutes, go right on over to video lesson 2C and watch it. As soon as it's over, then you can go to Schoology and take the exit slip on lesson 2B and 2C. I hope that's clear for everyone.